Our our topic is high pressure liquid chromatography, and we are lab group 4. So, what is high pressure liquid chromatography? High pressure liquid chromatography, also known as high performance liquid chromatography, is a separation technique for compounds of low or no volatility. And so, volatility is just the tendency of a substance to evaporate at normal temperatures. The separation technique is based on the partitioning of sample components on a stationary and mobile phase. The mobile phase is a lot more variable than stationary phase, as you can change the pH and the polarity of the solvent used. This highly improved form of column chromatography forces a solvent under pressures of up to 400 atmospheres instead of it just letting it drip down a column as normal. So here is a picture of a typical HPLC system with everything being labeled, such as the fluorescence detector, the solvent manager, the sample manager, the column chamber, and then the graph called a chromatogram. So more on the technique is that this technique allows much smaller particle sizes for the column packing material, which gives a greater surface area for interactions between the stationary phase and the molecules flowing past it, which this technique allows for a much better separation of the components of the mixture. And there are two variants depending on the polarity of the solvent in the stationary phase. One is called the normal phase HPLC, and the other one being reversed phase HPLC. And this diagram basically just shows how HPLC works going through the steps. So what is normal phase HPLC? This column is filled with a small silica particles with the solvent being nonpolar. A common nonpolar solvent example would be hexane. The typical column has an internal diameter of around 4.6 millimeters and a length ranging between 150 to 250 millimeters. A nonpolar solvent is used in this form of HPLC due to the particles being able to pass quicker. If a polar solvent would have been used, the particles would stick to the polar silica and slow the process much more. So this little diagram here basically just reinforces that the stationary phase is nonpolar while the mobile phase is polar. Another thing about the normal phase HPLC is that although it's called normal, it doesn't mean that it's most commonly used, because this is actually not the most commonly used form of HPLC. So what is the most commonly used form of HPLC? And that would be the reverse phase HPLC. It is similar to the normal phase in that the column size is the same, but the silica is modified to be the nonpolar in this form. So what they do, they modify the silica particles by adding long hydrocarbon chains to it. And these hydrocarbon chains usually range from about 8 to 18 atoms. So due to this, a polar solvent will be used this time. An example of a common polar solvent used would be a mixture of water and an alcohol such as methanol. The hydrocarbons added to the silica keep the attractions to a minimum, which causes the polar molecules to spend most of the time moving with the solvent. So if a nonpolar solvent had been used in this form, they would have formed attractions with the hydrocarbon chains that are on the silica particles through van der Waals forces. They would also be less soluble in the solvent due to the need to break the hydrogen bonds and squeeze in between the water and methanol molecules. So this diagram here just basically shows that the mobile phase is polar while the stationary phase is nonpolar in a reverse phase HPLC.
So next, I'll talk about the basic functions of the HPLC. So the HPLC's main method of operation is based off the principle that compounds act differently when put into water. The HPLC separates compounds into their components and purifies them. They are separated by their polarity, which is the link to how they behave differently in water. And before I talk about the process of HPLC, I must introduce the components of HPLC, and those being HPLC column, the HPLC sample/injection in the mobile phase, and then HPLC run. So first with HPLC column, This is considered the workhorse of the HPLC, and it consists of silica, which functions due to the long carbon chains, as mentioned before, and is often an 18 carbon chain, which can be called C18. So next, being HPLC sample injection. The HPLC sample types can vary depending on what's being tested. So anything such as urine, blood, saliva, drugs, toxins, it's a very broad range of the samples that are injected. But usually the samples get injected into a column via a machine. But a fun fact is that before this machine was created, that a person used to have to manually inject into the column. So they would have to sit there throughout the whole time that the HPLC would run. And it was a very taxing and long time. So thanks for the invention of the machine. And then the mobile phase is an organic solvent that moves through the column that flushes out compounds from the column. And this is a creation of the concentration gradient. And lastly, the HPLC run. Most common, as mentioned, is the reverse phase HPLC, and the run is basically just the whole process, and usually the run takes anywhere between 10 to 60 minutes. It's a very broad range. So now that I've introduced you to the components, I can say what happens. So first, the sample is injected into the column with many different compounds being injected at separate times. So many things can be tested at once, like multiple different samples are put in, not just the same sample. And then the mobile phase is then pumped through the column by a motor. And the addition of a solvent creates this concentration gradient, which decreases the polarity of the mobile phase. And the compounds stick to either the column or the mobile phase, depending on the polarity. But eventually, all compounds will stick to the mobile phase when their polarity matches with the polar phase. And then they disassociate from the column, and they all exit the column at a particular time. This time is known as the retention time. So basically at the end of the HPLC, you will see a graph, and this is called a chromatogram. And basically this shows the RF value, which is also known as the retention time. And this shows basically when each thing exited, or the time that it exited after the end of the HPLC process, once the mobile phase washed it all out. And as you can see in this graph right here, there's a ton of peaks, and each peak is representing basically those times of when they were gone out and all of that.
Performance of the HPLC. Compared to other chromatographic alternatives, HPLC is quick, easy, and requires little skill. The efficiency of this device is due to the fact that it uses a pump rather than gravity to force the liquid solvent into the adsorbent metal. The entire process takes only 10 to 30 minutes to receive results. The automation of this device accounts for not only the speed, but also the ease of use. The process of this device is highly automated, so little work has to be done to receive results. People who use this device require minimal training. The process of using this device is also extremely accurate due to the use of the pump, which makes everything much more precise and clear when reading, since there's not much room for human error. Despite the speed and accuracy of this instrument, HPLC is not always right for every situation. For example, high-pressure liquid chromatography is ineffective at separating volatile materials, as those are better off being separated by gas chromatography. Certain substances are also unable to be separated by high-pressure chromatography. These substances can be completely and irreversibly adsorbed by the metal and thus unable to be detected by the device. Though the device is easy to use due to the automation of it, there are still many complicated steps within the instrument. This makes using high-pressure liquid chromatography very costly, as well as difficult to troubleshoot if something within the instrument malfunctions.
Our first source is called the Multi-Detection of Antibiotics in Liver Tissue by Ultra-High Pressure Liquid Chromatography Tandem Mass Spectrometry. This specific type of HPLC used here is a technique that combines the separation capabilities of liquid chromatography with the mass analysis capabilities of mass spectrometry. So why this instrument? This instrument was used because they wanted to expand the tools that could be used to identify antibiotics in liver tissue. There are relatively few multi-detection and multi-class methods for the determination of antibiotics in liver tissue, so they wanted to create an experiment to discover if this type of HPLC could also be used for the determination of antibiotics in liver tissue. What type of sample did the author work with? Liver tissue is added with internal standard solution and chemicals for mobile phase. The internal standard solution was to represent the antibiotics in the liver tissue. There were 6 internal standards that were used in this experiment. One more well-known example of the internal standards is penicillin V. Penicillin is a common antibiotic we have seen used as a treatment for pneumonia, strep throat, or meningitis. There were also chemicals for the mobile phase added to the sample. Some of them include methanol, hexane, and formic acid. How was the collected data presented in the literature? The collected data was compared by using 2 different tables seen here. The first table is the point of reference of the current and accurate detection system used at the time for antibiotics in the liver. The second table was to represent the validation procedure that was performed where they analyzed 20 blank liver samples from different animal species and spiked them all with the compounds of interest at the validation level to prove the identification capability of the HPLC method. Some important highlights of this experiment are the proposed method allows to determine 39 antibiotics in liver simultaneously, and the present method is a huge improvement for laboratories involved in food safety control. And our second source is called Determination of BPAF in tissue, serum, urine, and feces of orally dosed rats by ultra-high pressure liquid chromatography, electrospray, tandem mass spectrometry. BPAF has broad applications in such areas as food processing equipment and electronic devices. There is a concern about the potential toxicity of BPAF. In this study, a simple method was developed for the determination of BPAF in various tissues and excretion of rats after an oral dosage was distributed. So why this instrument? This specific type of HPLC was used for the identification and quantification of BPAF. The HPLC would help to separate the BPAF in the tissues or excretion from the rats that was collected for easy identification. What type of sample did the author work with? Urine, feces, whole blood samples, kidney, liver, testes, and muscle tissues were dissected from the rats or collected from the rats. There were 8 rats in this experiment. 4 rats did not receive the dosage, and 4 rats did receive the dosage of BPAF. All the rats were used for observation and comparison over the span of multiple weeks in a controlled environment. How was the collected data presented in the literature? It was collected on a table with the separation of type of sample and whether the rat received the BPAF dosage or not. The data was clearly displayed with the concentration of BPAF in dosed and non-dosed rats. The difference between each is on the table for easy comparison of the concentrations. A highlight of this experiment is it was successfully applied to the determination of BPAF in tissues and excretion of orally dosed rats.
And here are our sources that we use during this presentation.